Hello, everyone. My name is Shanie. I'm an undergraduate at the University of Oregon. I'm majoring in biology and minoring in earth science. And today I'm here to talk about my research. Exposure to chemical signatures of fish predation increases survivorship in the invasive microcrustacean Daphnia lumholtzi. So, aging can be defined as the deterioration of physiological functions that affect survival and reproduction. It's known that environmental and genetic factors contribute to variation in aging and health span. However, this can also limit the identification of aging mechanisms. Many model organisms are constrained by these limitations, but Daphnia are a viable solution to isolate for genetic and environmental effects. And based on preliminary findings from a study done in our lab previously, we indicated that predation cues may actually increase lifespan in Daphnia lumholtzi, suggesting a complex relationship between predation pressure and longevity. So the big question was, are Daphnia who are exposed to predatory cues living longer than Daphnia who are not exposed? A little bit of background. So Daphnia can pick up on chemical signatures in the water that induce defensive phenotypic responses. So here we see Daphnia lumholtzi, which are known for their elongated head and tail spines. So whenever they detect pheromones in the water released by fish that feed on the Daphnia, these little guys then phenotypically respond by growing head and tail spines. Usually this is induced in early life stages and their offspring. And also, asexual reproduction is a mode of reproduction that the Daphnia use. And this is wonderful because this helps contribute to our experimenting in laboratory settings. This means that the Daphnia are able to provide us with genetically identical replicates, helping us to isolate for any environmental and genetic influences that I mentioned earlier. A mature female can produce roughly 4 to 8 clonal replicates every few days until she expires. And they usually live to about 30 days or so. So to start off, we begin with a common garden rearing experiment where we raise a select number of Daphnia, or mothers, over 3 generations in a controlled environment. This allowed us to minimize any maternal effects on our experimental offspring. And while we were conducting the common garden rearing experiment, we prepared our artificial lake media. The experimental media was conditioned for 3 days using 3-spined stickleback, which were fed Daphnia daily to ensure that the pheromones were getting released into the water. The media was then filtered to get rid of any matter, as we only wanted to test the effects of the pheromones. And the control media was pretty much prepared the same way. It was just absent of any fish, thus absent of pheromones. And Daphnia are filter feeders. Their primary source of nutrition is algae. So we cultured and grew our own algae, Cenodesmus obliquus and Chlorella vulgaris, in the lab. And once the algae was mature, we spun it down and diluted it to a density of 20 micromilliliters using a Secchi stick in the artificial lake media. So once we got through our 3 generations of the common garden rearing experiment, we pulled our 4th generation, the experimental generation, from those moms. For this, we had to do collections every other day because the moms could only produce so many neonates per day. So collection 1 was conducted on the first day of the trial, where we collected 36 for the control group and about 35 for the experimental group. And then every other day, we would continue pulling samples until we had a suitable sample size for our experiment, which totaled to about 375 individuals in the end. So in total, it was about 5 collections. Daphnia were then sorted into their own individual 50 milliliter beakers, where we made sure that they had 30 milliliters of either their control or experimental artificial lake media. Every other day, the Daphnia were checked for mortality and reproduction, and simultaneously, they were transferred to clean beakers with fresh media and were fed approximately 5 milliliters of that algae mix. This procedure was followed all the way up until every single Daphnia had expired. And we ran data analysis of our recorded data using survival packages through R. And this got us our beautiful Kaplan-Meier curve here. This shows us that within the first 10 days, we see here, the control neonates exhibit a significant decrease in survivorship compared to the predator-exposed groups, and that our predator-exposed groups continue to express higher rate of survival until the end of life. And down here, our hazard ratio was calculated to be 0.5 with a confidence interval here. This tells us how confident we are in our hazard ratio. So this hazard ratio confirmed that the predator-exposed Daphnia had a lower hazard of death compared to the genetically identical control Daphnia. And this indicates that the probability that the predator-exposed Daphnia are alive at any given time are 0.5 times higher than the control. And we also have a really small p-value, a p-value of below 0.001. And we know any scientist loves a small p-value. So this was absolutely wonderful to see. This indicates that the difference in mortality rates between the populations is statistically significant and unlikely due to chance. So in conclusion, we confirmed that the clonal replicates of Daphnia lumholtzi who were exposed to those chemical signatures expressed a significant increase in survivorship. These findings also suggest that survivorship is life stage dependent. As I mentioned before, we see this significant change within the first 10 days of life, where the exposed Daphnia have that higher survival probability. Though we were unable to prove increased end-of-life expectancy, the experimental Daphnia had consistently maintained that higher survival probability throughout the duration of the experiment. And really quick, I just want to thank Shannon Snyder and William Kresko for their mentorship and the rest of the Kresko laboratory team for their support. I also want to thank the UO Trio McNair Program Director, Dr. Christabel Dragoo, for her endless support and encouragement, and the rest of the Trio McNair Scholars Program for funding my research and all of the resources they have provided along the way to help make this possible for a first-generation low-income college student. Thank you so much for listening, and I hope you all have a wonderful day.